It gives me great pleasure to introduce the last speaker of the session today before our two lunch panels. And that, of course, is Bill Sellers. Bill, I think, needs no introduction for many of you. You know him, particularly if you're in the Boston area, from what was his former position as Vice President and Global Head of Oncology at Novartis, at NIBR, actually, Novartis Institutes for Biomedical Research. Bill purports to be retired now, but I can't imagine a scientific mind as fantastic as this one is actually retired. So I think you're going to hear some really interesting thoughts from Bill in his talk today. And the title is From the Identification of Cancer Vulnerabilities to Therapeutic Development, and it is not present. Oh, it's because you're asleep.
Thanks for inviting me, and I'll just say one other thing. I'm from a long family of Tufts alumni. My father, my mother, my sister, and most recently my son in 2015. And I'm sure you all know my son because you probably are all at the Tufts hockey games watching him play hockey. That's a joke because only family members were ever at those games. So I'm quite the reductionist. Even though I like high throughput biology, we tend to apply it in a very reductionist way. And the two questions at Novartis we were most interested in asking are, what are the genes that are required for the maintenance of the cancer phenotype? And I use the word maintenance specifically, despite the fact that it might be better to prevent cancer or stop its initiation or block its metastatic process. The 99.999% of patients being treated today are being treated because they already have cancer, and we need to try to eliminate it. And then the second question, which I think our first speaker, our last speaker got at, is how do we drug these? And I would share his enthusiasm for going beyond what is considered the sort of druggable set of genes as we know them today. So over about the last 10 years, and Dave Ruddy is here in the audience who's experienced every one of our transitions along the way of trying to discover cancer essential genes through the use of first siRNA and shRNA. Most recently, we've been using, like many others, pooled shRNA screens to try to identify those genes that are critical for cancer maintenance. And the way we do this is to synthesize large pools of shRNAs on chips to clone those into plasmids, use those plasmids to generate lentiviruses. The lentivirus pool is then used to infect a cancer cell line. That cancer cell line is then allowed to double over time, and at the end, we count the abundance of each shRNA in the population by next-gen sequencing. And so you can imagine, this is just a schematic diagram of how this might work. At day 0, we imagine the abundance of all shRNAs is relatively equal. Actually, it's not equal, but there's a distribution. And that distribution is going to change based on the effect the shRNA has on cell viability. So if, as in this middle red column, the viability of a cell is adversely affected by an shRNA, its sequence will be depleted from the population, and the abundance of the sequence in the next-gen sequencing output will go down, versus in green, where this shRNA would have an enhanced effect on cell viability and its abundance would grow. And of course, the vast majority, really, we think, are not going to change because they're not going to have an effect on viability. Now, as I said, we've been doing this for a long time, and some of our first attempts were to use small shRNAs against a small number of genes, so 5 shRNAs per gene, 1,200 genes in 250 cell lines, and I'll just summarize that by saying that data was too noisy, even though it was a lot of work. Then, together with our colleagues in the Development of Molecular Pathways group, we did a small pilot screen where we targeted 17 shRNAs per gene, really this being enabled by the sort of drop in the prices for oligonucleotide synthesis on chips, targeting 385 genes in the epigenome in 80 cell lines. And that data was really quite nice. We discovered BRG as a synthetic lethal with BRM, but the data set itself was too small. But the two experiments together led us to conduct something that we called Project Drive, for deep RNA interrogation of viability effects in cancer, where we targeted about half the genome, 7,500 genes with 20 shRNAs per gene, and we did that screen across 390 cancer cell lines. So how do we take those 20 shRNAs and convert them to an output at the end? Of course, each shRNA has its own pattern of on and off target effects. And one underlying assumption is that the off target effects will be relatively non-coherent because they'll be off target, and that the on target effects would be relatively coherent. And with 20 shRNAs per gene, you'd be able to identify a coherence among sets of shRNAs that reflected the actual on-target effect of depleting a certain gene product from the cell. And so using an algorithm developed by the Broad Institute called ATARIS, we take the pool of shRNAs and create a gene score for each gene in that population for each cell line. And then that ATARIS score is used here in a statistical model to show and plot the effect on cell viability for depleting that gene. And here I'm showing you the concordance of BRAF viability effects with BRAF mutation. You can see the concordance is quite strong. So this is just a plot to show you what it's like to go from the 20 shRNAs and why you might worry about just using 1 in your lab or 10 or 5. This is all of the shRNAs against KRAS against every cell line. And I think you'd be hard-pressed to go in there and pick out 5 that you might want to use as your definitive loss of KRAS experiment. But if you now convert that from each individual shRNA to the ATARIS score, and then color each cell line by whether it is or isn't KRAS mutation, you can see that the vast majority of cells whose viability is affected by KRAS are mutant for KRAS. This may seem sort of obvious, but at the time we did this in the literature, there were people going back and forth on whether KRAS dependence was uniquely associated with KRAS mutation versus other features of how you might activate a cell. So I'll show this. This can be somewhat controversial, but with this data, we can now go look at all of the publications of synthetic lethal effects with KRAS. So this is the data I just showed you now, shown as a little graph. And these are 5 publications reporting different genes that were thought to be synthetic lethal with KRAS mutation, all starting from a much smaller pool of shRNAs. And you can see in the larger data set, we're not able to recapitulate the dependence of cells on either the genes to the left on the KRAS mutation status. It's not to say the data are wrong in the paper, it's just that they're not reproducible across a large scale of shRNAs and across a large scale of cell lines. And I think these were largely false positives based on a small number of shRNAs being used in the original discovery. So here's an example of just one class. We've described sort of in our own work 4 or 5 different classes of genes that can come out of this large-scale analysis. The most obvious one is the class of genes that are oncogene drivers and hence have genetic dependence. And I'll just show you again BRAF, NRAS, PIK3CA, and then even FGFR2, which has only 2 or 3 cell lines that had FGFR2 alterations. The data are robust enough to pick those out as uniquely dependent on FGFR2. The second class of genes, and probably the largest class of genes that come out as specific eliciting specific dependence in certain cancers, are those that encode lineage-specific transcription factors. And I'll show you here MITF, which is the master regulator of the melanocyte lineage, something myself and Levi Garraway published on a long time ago, is uniquely required for melanoma in cancer. Similarly, SOX10, which is also in the melanocyte lineage, is uniquely required in melanoma. And in this case, because we also have the DNA methylation status for every cell line in the collection, we can specifically show that the reason SOX10 is required in these cell lines and not the ones shown in black is in these cell lines, the promoter of SOX10 is unmethylated, where in the rest of the cell lines, it's all methylated and off. So they don't care about SOX10 because they've never expressed it in the first place. We're able to recapitulate some other findings that have been previously published, for example, the requirement of MIB in the hematologic malignancy space, and then work from Miles Brown that showed FOXA1 was a specific breast cancer, hormone receptor positive breast cancer dependent gene. So for the rest of this section, I'll just talk about a discovery we made coming out of this data set, one of the novelties we were able to publish. And this involved the interaction between the arginine methyltransferase PRMT5 and a metabolic gene known as MTAP. And we're calling this a collateral synthetic lethal interaction. And you'll see sort of what I mean by this. This was published recently in Science. The first author is Mavrakis. So the reason we were interested in this metabolic gene MTAP, methylthioadenosine phosphorylase, is that it's been previously known, and people have looked at this in the past, that MTAP is closely located near CDKN2A, or also known as P16, on chromosome 9. And because of that, and because P16 is the target of deletion in so many cancers, MTAP is co-deleted along with P16 in a high proportion of cancer cells. So shown here in orange is the frequency of P16 deletion, or CDKN2A, and shown in yellow is the frequency of co-deletion. So you can see there are deletions that affect P16 that don't affect MTAP, but a huge proportion of those cell lines or cancers that delete P16 also delete MTAP. So what does MTAP do? Well, methylthioadenosine phosphorylase is an enzyme in the methionine salvage pathway that metabolizes methylthioadenosine. Now, I'll get to that in a second. And as part of its metabolism of methylthioadenosine, it eventually regenerates methionine, which together with adenosine and ATP generates SAM. And of course, SAM is an important cofactor for lots of enzymes that require methyl donors, including arginine methyltransferases. So we thought or hypothesized that if you delete MTAP, you might first predict that MTA levels should rise in cells. And there have been some published reports of this that we wanted to also confirm. So we took a number of cell lines and by mass spec measured MTA levels in those lines that had MTAP/P16 deletion versus those that didn't. And you can see MTA levels rise substantially in cell lines that have a deletion of MTAP. And I'll just draw your attention to this pair here where we used CRISPR to knock out MTAP in a cell line that had wild type MTAP and showed that the selective knockout of MTAP leads to a rise in MTA levels. So it's not just this big deletion that's doing it. You can recapitulate the rise in MTA levels by a specific knockout of MTAP. So the question is, if you go back to this, if MTA levels rise, if this is cut off, would you become dependent on other enzymes in the methionine salvage pathway? This was the original question. And so we looked specifically in our data set to compare those cells that had an MTAP deletion versus those cells that did not, to ask, were there shRNAs selectively being depleted from the population of cells that had an MTAP deletion versus those do not? And you can see one of the enzymes in the methionine salvage pathway actually did score MAT2A, which if we go back a second, is shown here. And we could hypothesize that if you don't have this, you're now not going to even use exogenous methionine in this pathway, and now you're dead because you have no way to generate SAM. But what was really interesting and a big surprise to us was that the most statistically significant finding was that PRMT5 was highly selectively lethal in MTAP-deleted cells. So what was also interesting in this data set, and I think this goes to having used so many shRNAs and having what we think is a really robust data set, is that we're able not only to find PRMT5, but the cofactors that PRMT5 uses to execute its function. So first, PRMT5 is found nearly entirely as a heterodimer with the WD40 protein known as WDR77. And you can see one of the top hits also was WDR77. So we pull out the protein complex in this analysis. That complex, in addition, forms two additional complexes, one with rheokinase 1 and nucleolin, and the other with a splicing complex known, encoded by PICIN. And both rheokinase and PICIN were found as hits. So this gave us really strong confidence that this protein PRMT5 plus its complexes were selectively required for the viability of cells deleting MTAP. So even though I would say that data was so strong, I wouldn't have needed additional validation to be excited, we did go on and do stable lines with DOX-inducible shRNAs to show that you could recapitulate this phenotype. So shown here is a DOX-inducible shRNA to PRMT5. We turn on the doxycycline and we get a knockdown. If we then overexpress a cDNA resistant to that shRNA by virtue of having silent nucleotide changes in the cDNA, we completely rescue the phenotype, suggesting that it's an on-target phenotype. Now, I'm not going to show you the data in the interest of time, but in addition, we showed that in those cell lines we made that were isogenic, where we CRISPR knocked out MTAP, they became sensitive to PRMT5 knockdown, so we know it's specific to MTAP. We also showed that using the DOX-inducible shRNA in an in vivo setting in a xenograft, we could get loss of viability in vivo and regression of MTAP-deleted tumor xenografts with PRMT5 knockdown. So the question we had, of course, is why is this? It didn't really make intuitive sense at the beginning, but if you look at the metabolite MTA, of course, it's a metabolism product itself of SAM, and hence they're structurally similar. And in fact, there had been previous publications that suggested accumulation of MTA might lead to inhibition of the PRMT5 family. So that was a clue that we followed up. And the way we followed this up is to test MTA itself, this metabolite that's accumulating in cells, to see whether it could inhibit PRMT5 or other methyltransferases. So we have a large panel of methyltransferases we tested here. We first tested them with a SAM analog, which is an inhibitor called sinafungin. And you can see that the panel is active. That is, we can detect inhibition of many methyltransferases with this non-specific inhibitor. But surprisingly, MTA inhibited only PRMT5. So for some reason, this metabolite, though it's an analog of SAM or very similar to SAM, inhibits only PRMT5. So we went on to solve the crystal structure of MTA and sinafungin bound to PRMT5, and this wasn't working so great the first time, but I hope it works now. So this is sinafungin, a SAM analog bound to PRMT5, and you can see it's making contacts with this lysine at 333 and a tyrosine at 334, and this is the arginine substrate sticking into the pocket that would normally be methylated by PRMT5. So, yeah, like I said, not working that well. So that's showing what I just told you. And now we're going to switch to the MTA bound structure, hopefully. Yep. So in purple is MTA, so I'm sorry the movie's not working so well, but you can see that it lacks this extension on SAM, and in the presence of PRMT5, this glutamic acid shifts positions here, E435, and now takes the place of SAM in coordinating the lysine at 333 and the tyrosine at 334. So when MTA is present in PRMT5, you get this reordering of the catalytic domain. And what's interesting about this is that lysine and tyrosine are only present in PRMT5 in this methyltransferase family and are conserved between Drosophila and human PRMT5, but are missing from the rest of the PRMT family. So we believe that this explains the specificity of MTA, though we have not done the killer experiment, which would be to take, for example, PRMT1 and convert this to lysine tyrosine, and now show that MTA can be a PRMT1 inhibitor. So that would be a cool experiment to do. So the question is, can we show in a reconstituted system that MTA can actually act as an inhibitor of MTAP? And we did this by first knocking down PRMT5, as shown here, and then rescuing with a cDNA for MTAP. So now we've reconstituted the cell with MTAP. MTA levels should have gone down. PRMT5 viability is erased. But if we add exogenous MTA, we now confer susceptibility to PRMT5 knockdown. So both MTAP deletion and MTA accumulation are required for the sensitivity to PRMT5. So our model for this is that under normal circumstances, PRMT5 is active. Under circumstances where you delete MTAP, MTA builds up and you get a partial reduction in PRMT5 activity that is then rendered a lethal reduction by the shRNA that we have applied to those cells. In fact, if you completely knock out PRMT5 by CRISPR, cell viability is affected in all cells. So this is one case where the shRNA experiment might in fact replicate a small molecule versus the complete loss of function, which would have told you this is just going to be a pan-essential gene. I'm going to skip that in the interest of time and switch to the second topic, which is our work on discovering allosteric inhibitors of the SHP2 phosphatase, and this was also recently published in Nature. So another interesting profile can be seen here in the DRIVE data set, which is if you look at SHP2 and you now color code the cell lines by whether they do or do not have an RTK driven phenotype, that is, they have an amplification, a translocation, or a gain of function mutation in an RTK, you can see that SHP2 has preferential loss of viability, not exclusively, but preferential in those cell lines that we classified by this method as RTK dependent. Conversely, if you look now and color those by RAS-RAF dependent, again, it's not perfect, but there's a redistribution of the RAS-RAF dependent cell lines now being insensitive to SHP2. Now that was exciting to us because of the way SHP2 is thought to function. So SHP2 is thought to be inactive in its normal state, held inactive by two SH2 domains. Upon binding to activated phosphorylated RTKs, it's thought that this binding event reorders SHP2's inhibitory effects of these two SH2 domains, creating an active phosphatase, and that in still an unknown mechanism triggers RAS pathway activation. Now, the evidence that this might be important in human cancer comes from the fact that there are mutations in SHP2 inherited in kids with juvenile myelomonocytic leukemia and sporadically or somatically in some solid tumors. And those mutations affect primarily the SH2 domains or the interface between the SH2 domain and the phosphatase domain. And those mutations, unlike, for example, PTEN, which is inactivated by mutations, are gain of function activating mutations in SHP2, suggesting that SHP2 is an oncogenic phosphatase. Now, we're also excited by work from Todd Golub's lab, our own group, and by Renee Bernard's. And to sort of very quickly summarize this, all of these data suggest that feedback bypass of various inhibitors, either RTK inhibitors or MEK-ERK inhibitors, is mediated through either growth factors like HGF, FGF, NRG, neuregulin, or EGF. And Renee's work in particular showed that these feedback loops are SHP2 dependent. So we're struck by the possibility that SHP2 might be a, let's say, universal mediator of adaptive resistance to other, in terms of either ERK-MAP kinase inhibitors or RTK inhibitors. I'm going to skip that in the interest of time. So we did the sort of typical thing that people do when they start drug discovery. They take the easiest thing you can make as a protein, which is the catalytic domain only. It's active. You don't need to worry about regulatory domains. And we did a screen, and we got nothing. Or we didn't get nothing. We got the same things everybody else got, which are cysteine reactive molecules binding to the active site cysteine. Now, to the team's credit, they decided to continue working and make the harder assay, which was to make a full-length SHP2 in vitro and activate this with a synthetic IRS peptide that had two phosphorylation domains, so that now the activity of SHP2 in this assay would depend on the peptide activating through the SH2 domains. And just to show you that how this worked is, if we take a measure of phosphatase activation and we now normalize that fold activation, peptide versus no peptide, you can see the truncated protein is not activated further by the peptide, whereas we get a robust activation of the full-length protein by the peptide. And this formed the basis of a screening assay, which was first used in not an HTS, but a pilot screen. So I know 100,000 compounds might seem to many people to be an HTS, but in reality, that's the pilot scale we do prior to HTS. And what was kind of cool is in the pilot screen, we of course have a lot of hits shown here, and this just shows you how noisy screens can be. All of these hits were subsequently revalidated in IC50 assays. 4 of those hits validated, including one called SHP836, which I'll show you the structure of. So SHP836 is shown here. It had an IC50 out of the primary screen of 5 micromolar. You can see here, it does not inhibit the catalytic domain only. It only inhibits the assay in which we're activating SHP2 using the doubly phosphorylated peptide, strongly suggesting this is not a pure catalytic inhibitor. Using that assay, further optimization was done, not a ton, to get to what we're calling SHP099 in the publication, which is a 71 nanomolar inhibitor of the peptide-stimulated SHP2. So how does this work if it's not binding to the catalytic site? Well, obviously it has to have something to do with the SHP2 regulatory domains. And I'm hoping this movie works better. So shown in green and blue are the 2 SH2 domains that I showed you earlier, and flying in from the left is the small molecule, and you can see it binds in an interface between the 2 SH2 domains and the SHP2 catalytic domain, which is all shown in brown. Now, you'll see that the positioning of the second SH2 domain, or the N-terminal SH2 domain, is such that there's a loop. It would be interesting if there's a loop from our prior speaker that, in fact, occupies the catalytic domain in this conformation and blocks access of substrates to the catalytic domain. And what's particularly cool is if you look at the interaction of this molecule, it actually makes hydrogen bonds with all 3 of the different domains, the 2 SH2 domains, and the catalytic domain, and we're sort of calling this a molecular glue for SHP2. So does this validate the genetic experiment we showed in the shRNA experiment? We haven't profiled this against that full panel of cell lines, but in leukemia cells shown here, 70 different leukemia cells, those that had RTK alterations such as JAK2 or BCR-ABL were sensitive to SHP2 inhibition versus those that did not have such alterations. And similarly, in colorectal cell lines, which don't have genetic alteration, we're able to define EGFR dependence by sensitivity to the EGFR inhibitor lapatinib versus insensitivity. And you can see there's a pretty good correlation between lapatinib sensitivity and SHP2 sensitivity, where those cells that are lapatinib sensitive are SHP2 sensitive, and the rest that are RAS-RAF altered have no effect of the inhibitor. We've also gone on to confirm that this molecule is active. It has good PK. It's active in vivo setting. This is a head and neck cancer, which has EGFR amplification and is sensitive to erlotinib. SHP099 and erlotinib have a comparable level of activity in this head and neck cancer tumor. And then this is a FLT3 primary AML orthograph, which you can see is extremely sensitive as a single agent to the SHP2 inhibitor. I'm going to skip this in the interest of going to lunch. And just summarize this part. The SHP2 is an oncogenic phosphatase. It's required for oncogenic signaling, mediated through RAS pathway activation. SHP099 is a novel allosteric inhibitor of SHP2 that induces the SHP2, the SH2-mediated inactive conformation of wild-type SHP2. This will not likely work against the JMML mutations that I mentioned before. In fact, that's a likely resistance mechanism. We've seen activity of this inhibitor in multiple RTK-driven cancers, including those of both solid tumor and hematologic origins. I'd just like to acknowledge a gigantic team of fantastic scientists at Novartis who I had the pleasure of working with for the last 10 years and whose interactions I miss dearly, though I have loved retirement so far. So far, yeah, winter might not be such a great time to be retired, including Dino Mavrakis, Rob McDonald, Mike Schlabach, Eric Billy, and Greg Hoffman, who led the whole Project Drive and the work around PRMT5, supervised by Nick Keene, Tobias Schmeltz, Francesca Hoffman, and Frank Stegmeyer. I mentioned Dave Ruddy for all his work in the shRNA drive project. And then on SHP2, Yingnan Chen, who led the biology, along with Homan Chan, Matthew Lamarche, who was the lead chemist for the SHP program, Tim Ramsey, who oversaw our chemistry group in Cambridge, Nick Keene, Travis Stans, and really Pascal Fortin, who also recently left Novartis, who really was the, let's say, father of pushing for the assay that led to the discovery of the allosteric mechanism. So thanks, and I don't know if there's time for questions, but I'm happy to take them.
Let's go ahead and take a couple of questions for Bill.
Hi, very impressive talk. I have two questions regarding the PMT5 project. So is the MTA treatment toxic to normal cells?
No, yeah, it's not. I mean, you would probably, because you can't achieve enough of PRMT5 inhibition. And the other reason you know it can't be toxic is we have cancer cells that have high levels of MTA, right? So if it had negative selective properties, the cancer cells would never have evolved the co-deletion of MTAP. They would have avoided, what would happen is you'd get selective deletion of P16 and you'd avoid the MTAP locus.
The second question is regarding the inhibitor. So I remember Epstein also developed a PMT5 selective inhibitor.
Yep. Mm-hmm.
Can you comment on this too?
Yeah, I didn't go over this because it often ends into a longer discussion. But so if you remember what I showed you, MTA is binding in the same site as SAM. So in the cells, you actually have SAM and MTA simultaneously. It turns out even though MTA is high, SAM is even higher. The epizyme compound is a SAM cooperative inhibitor. It requires and binds cooperatively with SAM to PRMT5, which means once that happens, the relevance of MTA levels in the cell become essentially irrelevant. So the PRMT5 inhibitor acts more like a CRISPR reagent and in fact is a sort of null for PRMT5 and doesn't distinguish between MTAP deleted and MTAP wild type cells. So you'd say, oh, well, how are you going to make an inhibitor? Well, we believe you need an inhibitor that's not SAM cooperative. So the enzymology is more complex, but we want a SAM agnostic inhibitor, or you could imagine MTA cooperative inhibitor would ideally take advantage of the high levels of MTA in the cell. And believe me, the reviewers had a hard time understanding this, I think because they're not enzymologists, but yeah, that's the enzymology. And in the paper, there's all the data around the epizyme inhibitor. It's all there.
Yeah. Okay, one last question back here.
Bill, can you comment on whether CRISPR has completely supplanted this pooled shRNA screening, or the pros and cons thereof?
Yeah, so the answer is yes for us. We had some debate about whether we should finish the next 7,500 genes by shRNA, and that's when all the CRISPR work was coming out. We've published recently our comparator in the first 5 cell lines between large-scale CRISPR and large-scale shRNAs, and we did a lot of work to eliminate false positives, but we had no idea what the false negative rate of shRNA screening, and it's substantial. And so with CRISPR, you pick up a significant number of hits that you don't find by shRNAs in just the first 5 cell lines. CRISPR itself has one notable off-target effect, which is copy number gain creates additional CRISPR cuts, which lead to toxicity that is gene independent. So when you scan across an amplicon, it looks like every gene and every region of the amplicon is cancer essential. This is also true if you have a CRISPR that happens to, by chance, cut 8 times in the genome. It will be 8 times more toxic than something that cuts once. So that's the downside of CRISPR, but the upside is significant gain in lowering the false negative discovery rate. And the false positive discovery rate is also pretty low, too.
Okay, thanks. Thank Bill.